Hello and welcome to Down the Scope. Today we'll be looking at the structure and function of shark kidneys. Many of you will be familiar with the function of mammalian kidneys as blood filters, in that they remove potentially toxic waste products such as urea from the blood. They also have a role in controlling ions such as sodium and calcium, as well as fine-tuning blood pH and water content. They are all singing, all dancing filters and reabsorbers. In many ways, the overall function of shark kidneys is the same, to maintain ion homeostasis, but they do this in quite a different way, mainly by maintaining urea in the blood rather than excreting it. Most sharks live in salt water, a fluid with high osmotic potential. If you'll bear with me while I bore you with some biochemistry. The osmotic potential of a fluid is based on the number of things dissolved in it, with this being measured in osmoles. Salt water has lots of sodium and chloride ions dissolved in it, giving a high osmotic potential. The rule is that if two bodies of water are separated by a semi-permeable membrane, and one side has a higher osmolarity than the other, then water will move from the area of low osmolality to that of high osmolality until an equilibrium is reached. Shark blood is often in close contact with salt water, separated by a semi-permeable membrane, that of the pillar cells. Consequently, if the osmolality of shark blood were lower than that of salt water, then water would leave the shark until the sea was dilute enough to stop this, or until the shark died of dehydration, the latter being a more likely outcome. So, sharks have made their blood slightly hyperosmotic compared to seawater. Shark blood has an osmolality of roughly 1000 milliosmoles per kilo of water. Compare that to human blood, which is around 290 milliosmoles per kilo of water. Sharks maintain such a high osmotic potential in the blood by using urea as an osmolyte. To achieve this, the shark liver pumps out prodigious quantities of urea, with urea production also found in other organs, such as the muscle and the stomach. But production is one thing, tight regulation is another. What if the salinity of the water changed? Then the shark's blood would be far too hyperosmotic, and water would begin to leach into the shark, causing all kinds of problems. Control of urea levels occurs in the kidney, both through retention and excretion. Equally, the renal filter system is not selective. This is true in mammals and other vertebrates. This means that after the filtration part, all organisms have developed elaborate systems of tubules lined by specialist cells that can then reabsorb all of the useful molecules that the filter carelessly threw out. In this way, shark kidneys reabsorb more than 90% of the urea present in the primary filtrate. The kidney of sharks is located against the dorsal surface of the caudal coelomic cavity. Each kidney is made up of irregularly shaped lobules. Here we can see that each lobule is further divided into two zones, called the sinus zone and the bundle zone. The filtration mechanism is the renal corpuscle, which separates the two zones. Of the two zones, the sinus zone is the largest in volume. It's characterized by renal tubules, as you can see here and here, separated by vascular spaces, such as this one and this one. The bundle zone is made up of renal tubules packed tightly together. Let's focus on the renal corpuscle for the moment. The corpuscle has two parts. The glomerulus, which is formed of a tuft of capillaries from which urine is filtered, and the Bowman's capsule, this large empty space here, which wraps around the glomerulus and collects the urine. You can clearly see that the epithelial cells of part of the Bowman's capsule, that closest to the tubules, also known as the urinary pole, are ciliated, which you can see as these wispy material here. The cells of the vascular pole, closest to the glomerulus, are squamous, meaning that they have a flattened appearance. There's a lovely scanning electron micrograph showing these in a paper by Lacey, a link to which is in the video description. In this image, which shows the junction between the two types of epithelium, you can see that the squamous cells have a single cilium on their surface. Over the glomerulus there are three layers: the endothelium of the capillaries, a thin supportive membrane called the mesangium, which is likely this darker staining material, and podocytes, which form the epithelium of the Bowman's capsule. These nuclei here probably belong to podocytes. The endothelium is fenestrated, meaning there are small pores or windows up to 70 nanometers wide, which allow fluid and small molecules to pass through. The podocytes extend finger-like projections across the capillaries, which branch and interdigitate, forming gaps of a similar size to the endothelial fenestrations, again allowing passage of the filtered fluid and small molecules. In this electron micrograph, you can see the podocytes lining up along the capillary in rows, while this image is a close-up of the pedocells, which branch up to 3 times and interdigitate over the capillary surface. After the corpuscle comes the nephron. The shark nephron is extremely long and has a more complicated structure than that of their mammalian counterparts. In mammals, the nephron makes a single loop down from the cortex into the medulla. In sharks, the nephron makes 4 loops. Starting from the corpuscle, the tubule enters the bundle zone and makes a turn back towards the sinus zone. This is the first loop. After a meander in the sinus zone, a second turn is made back towards the bundle zone, forming the second loop. In the bundle zone again, a third loop is formed, which is the longest of all, before the nephron turns last time back towards the sinus zone and turns back to the bundle zone, forming the fourth and final loop. From here, the nephron joins a collecting duct. All of this looping must have some functional significance, and the function and structure of the epithelial cells lining each part of the tubules is slightly different. As we can see from the schematic, within the bundle zone each nephron will have 5 tubules. These are all enclosed in a sac-like sheath separating a microenvironment within from that outside. And indeed, if we look at the bundle zone of the slide, you'll see groups of 5 tubules encased in a collagen sheath. Also within the bundle zone is a small lymphatic-like vessel called the central vessel, which we can see here and here. This originates within each bundle of tubules and flows towards the renal blood system. These anatomical loops can be divided into 5 functional regions. Starting at the renal corpuscle, there are the neck, proximal, intermediate, distal, and collecting segments. I've had immense difficulty differentiating these different tubule segments histologically. Maybe these sections aren't good enough, or maybe the differentiation is not as accentuated in younger animals. By cell morphology, we can only differentiate the proximal segment, as the tubules are lined by epithelium with a brush border, giving it a kind of fuzzy appearance. So this tubule here is part of the proximal segment. According to my references, we have to rely on size to help with tubule identification. Supposedly, the largest tubule in the bundle zone is the early part of the distal tubule, but many of these tubules are very similar in size, so perhaps we will have to leave it at that until I can get a better section of shark kidney. What is certain is that the 5 tubules here in the bundle zone are essential for urea absorption. At the moment, the model for urea reabsorption is hypothetical and incomplete, and best described using a schematic drawing. The current hypothesis goes something like this. In the early distal tubule, there is massive reabsorption of sodium and chloride. This increases the osmolality of the interstitium and draws water from an as yet unknown segment of the bundle zone. This generates a low urea fluid within the bundle. This low urea fluid enters the central vessel and flows towards the sinus zone. As water, sodium and chloride, as well as other solutes, are reabsorbed from the urine, the concentration of urea increases, forming a urea-rich solution which passes into the collecting tubule. The epithelial cells express urea transporters on both the apical and basolateral membranes, allowing reabsorption of urea from the concentrated urine to the low urea fluid in the central vessel. Some cartilaginous fish, such as the Potamotrigon genus or freshwater stingrays from South America, don't need to retain urea as an osmolyte because they don't live in salt water and consequently don't have a bundle zone in the kidney. So that's about all we can discern about the structure of the shark kidney and its role in physiology from these slides. In truth, I've not used the slides much, but they were an interesting starting point for a wider exploration of shark physiology. If you want to know more, I can recommend the article Morphological and Functional Characteristics of the Kidney of Cartilaginous Fishes by Susuma Hyodo which I used as a primary source for the information in this video. A link to the article is in the description. If you have any questions, leave a comment below and I'll do my best to find an answer for you. And don't forget to subscribe for more videos on the anatomy and histology of the dogfish and other animals. If you want to see slides of tissue from other animals, then you can visit the website. There's a link in the video description. Thanks for watching, and until next time, goodbye.